Dear students, welcome to Biology. In today's class, we are going to discuss the questions from the Unit 7, that is Genetics and Evolution. The questions asked in the year 2020 from the Unit 7. 11 questions were there. 11 questions were there from the 3 chapters: Principles of Inheritance and Variation, then Molecular Basis of Inheritance, then Evolution. So we can see one by one. The first question: the experimental verification of the chromosomal theory of inheritance was done by. We studied that the chromosomal theory of inheritance was put forward by Sutton and Boveri, isn't it? Sutton and Boveri had proposed the chromosomal theory of inheritance, but the question was the experimental verification. The experimental verification was done by Morgan. Okay, Morgan had crossed in the case of Drosophila. He had conducted some experiments. Then the chromosomal theory of inheritance was proved by the experiment conducted by Morgan. So Sutton and Boveri proposed a chromosomal theory of inheritance. Morgan proved the chromosomal theory of inheritance by conducting classical experiments in Drosophila melanogaster, in which he had proved there is a possibility of linkage and recombination, the parental combinations, and a possibility of non-parental combinations. So the right answer is Morgan. Now the question number 2. Second question: how many true breeding pea plants varieties did Mendel select as pairs? We studied that there are 7 pairs of characters, isn't it? 7 pairs of characters have opposite traits, 14 traits are there. So 7 pairs of characters. Which are the characters? So which were similar except in one character with the contrasting traits, opposite traits. So how many true breeding plant varieties? For example, if it is height, a true breeding tall pea plant and a true breeding dwarf pea plant. True breeding means homozygous, capital T and small t, isn't it? The homozygous condition is otherwise called a true breeding. So how many true breeding? For one character, 2 breeding varieties are there. So for 7 characters, 14 true breeding varieties are selected, isn't it? Next question. Question number 3. Identify the wrong statement with reference to the gene I that controls ABO blood group. The I gene is for isoagglutinin that control the blood groups. The ABO blood group. Which among the following is the wrong statement? When IA and IB are present together, they express same type of sugar. That is wrong. IA produces what? IA produces A antigen and IB produces B antigen. So they are different types. We studied that in the case of blood group, it is the inheritance pattern is codominance. No, IA allele and IB allele are equally dominant and both express simultaneously. IA will produce A antigen, IB will produce B antigen, so that the blood group will be AB, isn't it? So that is a wrong choice. Allele small I does not produce any sugar because small I is a recessive one. Then the gene I has 3 alleles. We know that phenomenon when a gene has more than 2 alleles, the condition is called multiple allelism. A person will have only 2 of the 3 alleles. Even though the gene has 3 alleles, a particular person will be having any 2 of the 3 alleles present. So the 2, 3 and 4 are right. The first choice is wrong. Okay. Question number 4. Select the correct match. Sickle cell anemia, autosomal recessive trait, chromosome 11. Thalassemia, X-linked. Hemophilia, Y-linked. Phenylketonuria, autosomal dominant trait. So in this chapter of Principles of Inheritance and Variation, we are not studying any dominant traits, dominant diseases. So all the diseases, we have studied 5 Mendelian disorders: color blindness, hemophilia, hemophilia, then sickle cell anemia, thalassemia, and phenylketonuria. These were the diseases, of which the color blindness and hemophilia were sex-linked or X-linked. Color blindness and hemophilia were X-linked. Sickle cell anemia, thalassemia, phenylketonuria, all are autosomal recessive. Here X-linked recessive. Color blindness and hemophilia are X-linked recessive. Sickle cell anemia, thalassemia, phenylketonuria, all these are autosomal recessive. So that means all the 5 genetical Mendelian disorders that we are studying in this chapter were recessive disorders. Myotonic dystrophy is dominant one. So color blindness and hemophilia X-linked recessive. So hemophilia Y-linked is a wrong choice. Phenylketonuria autosomal dominant trait is also a wrong choice. Thalassemia is not X-linked, it is autosomal. So that also is a wrong choice. Sickle cell anemia, autosomal recessive trait. Yes, autosomal recessive in the chromosome 11. The genes are present in chromosome 11. So it is the right statement. Choice number 1. Okay. So these were the 4 questions from the principles of inheritance and variation. 4 questions were then during 2020. Then we can move on to the next chapter, molecular basis of inheritance. So from this chapter, 3 questions were asked. Question number 1. Name the enzyme that facilitates opening of DNA helix during transcription. You see, transcription. So transcription means the synthesis of single-stranded RNA from double-stranded DNA. Isn't it? It is the RNA synthesis. The enzyme needed for RNA synthesis is called RNA polymerase. DNA polymerase is needed for DNA synthesis, that is DNA replication. DNA ligase is also needed during DNA replication so that the Okazaki fragments are interconnected by DNA ligase. DNA helicase is another enzyme, unwinding enzyme, that is also used during replication, not during transcription. So in transcription, RNA polymerase is used. So number 2. That is question number 2 from molecular basis of inheritance. Choose the correct pair from the following. Nucleases. Nucleases are enzymes which are needed for digesting the DNA or RNA. That means RNA breaking or DNA breaking enzymes. Nucleic acid digesting enzymes are called nucleases. So they are not needed for separation of DNA. They are for breaking of DNA. Exonucleases. Exonucleases are cutting the DNA at the outer areas. The peripheral cuts are by exonucleases. There are 2 types of nucleases, exonucleases and endonucleases. Exonucleases make peripheral cuts and endonucleases make internal cuts. So, make cuts at specific positions within the DNA, not within the DNA. Inside the DNA is endonuclease, specific position, cutting at specific position means restriction endonuclease. So, that is not the answer. The ligases. Ligases, as I already stated, ligases are enzymes needed to join the DNA fragments. They are ligature enzymes. So Okazaki fragments are interconnected by DNA ligases. Then exons are RNA fragments which are interconnected by RNA ligases. So nucleic acid joining enzymes are called ligases. Ligases join the 2 DNA molecules. That is a right option. Polymerases break the DNA into fragments. No, the nucleases are breaking the DNA into fragments. So the answer is 3. The right pair is 3. Ligases join the 2 DNA molecules. Okay. Now the third question from molecular basis of inheritance. Question number 3. The first phase of translation. What is translation? Translation means the synthesis of protein from RNA. Isn't it? RNA into protein. That process is called translation. What is the first phase of translation? For translation to take place, the amino acids are arranged in a particular sequence, isn't it? The sequence is given by the mRNA. Based on the base sequence of mRNA, the amino acids are arranged in a particular order. That process is called a translation. So for aligning arrangements in a particular order, the amino acids to be transported to the site of translation, right? Amino acids are present in different locations of the cell. So all these amino acids has to be transported towards the site of translation, and for the transportation there is another carrier molecule is there. The carrier molecule is called tRNA, transfer RNA. So at first we have to attach the amino acid with the tRNA. Attachment of amino acid with the tRNA producing aminoacyl tRNA. Producing aminoacyl tRNA is called aminoacylation, and that is the first step. So aminoacylation of tRNA is supposed to be the first phase of translation. So what is aminoacylation? The attachment of activated amino acid with the carrier molecule called tRNA, producing what? Aminoacyl tRNA. And this aminoacyl tRNA will be transporting towards the site of translation, and the amino acids are then released, and these amino acids are attached according to the mRNA codons, producing what? A protein of a particular function. Okay. It's a tricky question from evolution. The embryological support for evolution was disapproved by. Actually, the embryological support for evolution was proposed by Haeckel. Embryological support of evolution means the embryos of organisms are similar to the the previously formed organisms, isn't it? So that was proposed by Haeckel, Ernst Haeckel, but that was disapproved by the von Baer. Von Baer is the father of modern embryology, Carl Ernst von Baer. Choice 3. Question number 3. The flippers of penguins and dolphins are examples of. So you see, penguin is a bird, right? Penguin is a bird. Dolphin is a mammal. So both are having what? Flippers. Both are possessing flippers. So what must be the evolution called? It is convergent evolution, isn't it? Convergent evolution. So these are from different groups. One is a bird, other one is a mammal. So they are evolving in a particular direction so that they are producing an organ, and that organ is called analogous organ. These organs are structurally different but functionally similar. The functional similarity is called analogy, because the flippers are used for swimming in water both by penguin as well as by dolphin. But the structure of flippers in penguin and dolphin are entirely different. The anatomy is different. The origin is different. One is from bird, another one is from mammal. So that evolution is called convergent evolution. It is not radiation, natural selection, industrial analysis. It is convergent evolution is the right answer. Then the last question from evolution. Which of the following refer to the correct examples of organisms which have evolved due to changes in the environment brought about by anthropogenic action? Anthropogenic action means man-made action. Anthropology, study of human beings. So anthropogenic, man-made action. Darwin finches of Galapagos Islands, it is not man-made. Herbicide-resistant weeds, yes, herbicides are produced by human beings. Drug-resistant eukaryotes, yes, drugs are produced by human beings. Man created breeds of domesticated animals like dogs, also it is all man-made. So B, C and D are man-made, the evolution by anthropogenic action. B, C and D. Anthropogenic means man-made. Okay, it is not natural. So these were the 11 questions from the second unit of the second year, genetics and evolution. 11 questions were there. From principles of inheritance 4 questions, molecular biology or molecular base of inheritance 3, the evolution also 4 questions. Okay, thank you.